Now I'll study about the antibodies. What is antibodies? Polyclonal and monoclonal antibodies. So what are antibodies? Antibodies are basically uh, we'll see what antibodies are. Basically, these are glycoproteins. And what are antibodies? We'll see about the structure and function of the antibodies, and basically what is polyclonal antibody and what is the difference between polyclonal and monoclonal antibodies, and how how polyclonal antibodies are made, and how purification of the polyclonal antibodies is carried out, and what are the function of the polyclonal antibodies. We'll see what are the functions of the polyclonal antibodies and what are advantages and disadvantages of the polyclonal antibodies. So now let us see. So we'll discuss what is monoclonal antibody, how monoclonal antibodies are produced, and how the purification of the monoclonal antibodies takes place. What are the function of the monoclonal antibody, advantages and disadvantages, and difference between polyclonal and monoclonal antibody. What are its key applications and key points, and there are references also. Immunoglobulins is a protective protein produced by immune system, that is B cell, in response to presence of foreign substances. Suppose there is a bacteria and it is having some epitopes, then antibody will come and it will just stop it. It will just combine here. It will just come and it will just combine here with its structure, and it will try to attach here. So it will just stop it uh, in order to prevent it infecting the other cells. So antibodies are immunoglobulins, and it is protective protein produced by immune system B cells mainly in response to presence of the foreign antigens, and it is recognized and latch on the antigen in order to remove them from the body. Basically it latches on the antigen to remove it from the body. Now I'll see. So this is resting B cell. The resting B cell, there are membrane bound Ig immunoglobulins. In the resting B cell there are membrane bound immunoglobulins, and this membrane bound immunoglobulins basically encounter, suppose there is an encounter with an antigen, like antigen is a bacteria. So this is a bacteria, and suppose B cell encounters an bacterium, then this B cell stimulated B cell give rise to antibodies. The stimulated B cells give rise to antibodies secreting plasma cells. So these are plasma cells. These are plasma cells, and which are secreting antibodies. So this is the workflow, that is resting B cell encounters an antigen and it combines with the epitopes of antigen, and this stimulated B cells give rise to antibody secreting plasma cells. Now what is the mechanism? The most important mechanism to eliminate invader is by production of the antibody. So basically the production of the antibody takes place, and this is the most important mechanism to eliminate invader. inside the body. Now how this takes place? So basically an antigen binds to B cell surface and it stimulates B cells to divide and mature into group of identical cells called clones. So these are called clones. Matured B cell also called plasma cells, which secretes millions of antibodies into bloodstream and lymphatic systems. So in this figure you can see there is an antigen which can be a bacteria, and these are a special cell that eats antigens, and these are B cell, that is a lymphocyte releasing antibodies that attack antigen. So we are studying about antibodies, so we'll concentrate on B cells. So these are B cells or lymphocytes which basically is secreting antibodies which are attacking the antigens. Now what is the function of the antibody? The basic function of the antibody is it circulates and attacks the neutralizes the antigen, which activates the immune system. Antibody attack antigens by binding to them, and neutralization. What is neutralization actually? When antibody binds to a toxin, it neutralizes by poison by altering its chemical composition. Such antibodies are called antitoxin. Suppose there is a toxin produced by bacteria. And the antibody will just come and bind on it. When antibody will bind on it, it will just neutralize it. So in that case, we'll call it as antitoxin. An example is diphtheria. So diphtheria toxin is an example of it. As you can see, diphtheria is an example of this kind of toxin produced by the bacteria. Now you'll see, by attaching with the invader microbe, it can make microorganism immobile or prevent from penetrating body cell. Now another important concept is opsonization, the coating antigen for more phagocytosis. So basically here the bacteria or any other antigen, suppose this is a bacteria, and other antigen, suppose this is a bacteria, and it is basically coated with antibodies. It is basically coated with antibodies. And it causes the phagocytosis of that bacterial cell. So this is basically called as opsonization. Now what is activation of complement? It is a series of reactions actually. Basically it triggers the lysis, bursting of the invaders, that is antigen, and attract invaders. So basically it is a series of reaction where lysis or bursting of the invaders that attract microbe killing scavenger cells that ingest or phagocyte the invaders. So what is antigen receptor? For the production of antibody, it is important that B cell bind with the antigen, because after binding with the antigen only, it can carry out further process. Each B cell bind only one type of antigen and recognizes countless number of antigens. So B cell surface contains antigen specific receptors, that is antibody protein that recognizes the antigen. Particular B cells have identical antigen receptor, but on other it differs. So structure variation among antigen binding sites allow different B cell to recognize different antigens. So there are structural variation among, suppose there is an foreign invader bacteria, and it will have different kind of antigens on it, right? It will have different kind of antigens. As you can see, I am making with different colors. It can have different kind of antigens. So how this basically how this is recognized by antibodies? So this is because structural variation among antigens binding sites allow different B cell to recognize different antigens. Now, due to binding between receptor and epitope, antigenic determinants. So what are epitopes? This is also important to understand. So suppose this is a bacteria. This is a bacteria, and it is having different kind of antigenic receptor. For production of antibodies, it is important that B cell bind with the antigens. Each B cell binds Each B cell binds only one type of antigen, and it recognizes countless number of antigens. So B cell surface basically contain antigen specific receptors that bind and recognizes antigens. And particular B cell have identical antigen receptors, but other it is different. So the structural variation among binding sites allows different B cell to recognize different antigens. Due to Now, what are polyclonal antibodies? A polyclonal antibody is collection of many immunoglobulins, each generated from a different B cell clone. These antibodies target different epitopes. So these basically target different epitopes or binding sites of antigen or on a single antigen. So polyclonal antibodies are heterogeneous mixture which are usually produced by different B cells clone in the body. They are recognized and bind to many different epitopes of a single antigen. Polyclonal antibodies refers to a mixture of immunoglobulin molecules and which are secreted against particular antigens. Each antibody recognizes different epitopes. So this is the main characteristic of the polyclonal antibody. This means polyclonal antibodies are heterogeneous mixture of antibodies. These are not same. These are heterogeneous mixture of the antibodies. So this beautiful figure shows how polyclonal antibody works. So basically this recognizes different kind of epitopes present on an antigen, as you can see in the figure. So suppose there are different kind of epitopes, and this is recognized by the polyclonal antibodies. So these polyclonal antibodies are very less specific in nature, but it is very sensitive. That is the point. Specificity is very less, but sensitivity is very high. Now, how is the production of polyclonal antibody takes place? All antibody generation polyclonal are with the elicitation of an immune response termed as immunization. So basically this is done by the immunization process. The intended targets, that is host like mice, rabbit, whatever host you are choosing, recognized by the immune system as foreign and targeted by antibodies for immune blockage or clearance. Failure to mount an immune response would require the use of the adjuvant. Suppose you are injecting particular rabbit or some other organism with an antigen of your protein, and if immune response is not generated, you will need an adjuvant to amplify that immune response. Now immunogenic substances, these are like friends adjuvants or keyhole limpet hemocyanin to further derive immune recruitment. Immunization comprises the majority of what is required to make polyclonal antibodies. Now, general steps for production of polyclonal antibodies. Polyclonal antibodies are often produced by injecting lab animals like mouse, rabbit, goats with specific antigens to develop this antibody. We inject the antibodies inside our organism to begin the process. If after a few weeks or months, after 6 weeks, the immune system of animal will start producing the high level of antibodies specific for the antigen. The plasma cells present inside the body of that animal start producing antibodies. Then we extract these antibodies from the body of that animal to purify the antibody. Normally, in this case, the antibodies which are produced are mainly IgG along with the variations. Now this is the beautiful figure which shows how this antibody production workflow takes place. So here we inject the organism, inject antigen into the rabbit, and second, the antigen activates the B cell, and the plasma B cell produce polyclonal antibodies. Basically, polyclonal antibodies will be produced here, not monoclonal. Monoclonal is very difficult to produce. It involves the use of the hybridoma technology. Now these antibodies are screened, and the antiserum from rabbit containing polyclonal antibodies is obtained, and this contains our desired antibodies. So this is the production of the polyclonal antibodies. Now initial exposure for prime immune response. Initial exposure of the antigen with appropriate adjuvant exposure is termed as prime immunization. Typical method of exposure include injection into the skin, peritoneum, subcutaneous layer, or muscles. Less common alternatives are oral and intranasal. Now, DNA immunization is also an emerging technology for the toxic or otherwise difficult to produce antigens. Any subsequent exposure to antigen is termed as boost immunization. Antigen processing by antigen presenting cells. Suppose there are antigen presenting cells like B cell, dendritic cell, and macrophages. So they internalize antigens by endocytosis or phagocytosis. Antigen processing breaks down these antigens into smaller pieces. So inside the cell, this cell basically phagocyte the antigens, and there is breakdown of these antigens inside the cells, which and then they are moved to the surface. And they are presented as a antigen. That's why these cells are called antigen presenting cells. Now the stimulation of the B cells takes place. Recognition of the antigen presented on the antigen presenting cell induces the release of the signaling molecules, cytokines and helper T cells. This cytokine activates B cells which are recognized antigens through B cell receptors. Now some B cell do not require helper cells again. Helper T cell activation after B cell recognition interaction, usually producing a quicker immune response with lower affinity antibodies. The involvement of helper T cell is critical to replicate immune response as well as regulating self-tolerance. Now proliferation of B cell antibody segregation. Activated B cell divide to create both memory B cells and plasma cell population. Both type of cells recognize the same epitope. Now this is the workflow where white blood cell is there, then there is a lymphocyte, and there is a B cell. B cell produces plasma cell, and this produces antibodies, and this lymphocyte, again natural killer cell, this activates the T cells. This is the development of plasma cell. Now plasma cells secrete antibodies into the serum for immune recognition, while memory B cell persist for longer periods, providing quick response. So this is the difference, quick response, exposure to the same antigen. Now what is important is how these antibodies are purified. Purification of these antibodies. So steps basically involves combination of chromatography, precipitation methods. One can purify antibody with good yield and resolution. Why to purify polyclonal antibodies? Because basically they need to purify in order to remove all the possible contaminants like albumin, transferrins, and cell degraded products like DNA and cellular proteins. Now purification methods. The choice of purification method is generally based on the factor mentioned below: nature of antibody and how this nature of feedstock, scale of production, economic cost, and other factors, processing timing, and desired purity. Now process of purification. The process is basically multi-step process which mainly include the sample preparation, capture, initial purification, secondary purification, polishing, or formation. So what are the advantages of polyclonal antibodies? So polyclonal antibodies are basically inexpensive, quick to produce. Purified antibodies ready to use, used under months. Now easy to store. This is easy to store, and highly stable and tolerant to pH buffer changes. High overall antibody against the antigen due to recognition of multiple epitopes. Superior antibody affinity generally results in quicker binding to target antigen, ideal in assay requiring quick capture of the proteins such as IP and CHIP. So significantly more robust when assaying protein that show slight variations in individual epitopes such as denaturation, polymorphism, conformation changes. So superior for use in detecting native protein in multiple assay types. So see, these are the advantages. Now much easier to couple with antibody levels, less likely to affect binding capability. What are the disadvantages? Disadvantage is basically the variability between different batches produced in different animals at different times, higher potential for cross reactivity due to recognizing multiple epitopes, and affinity purification of the serum will typically be required to minimize the cross reactivity. Basically, there's always chances of cross reactivity in the polyclonal antibodies. So what are the uses of polyclonal antibodies? Polyclonal antibodies are basically used to experimentally and in clinical medicine for a variety of reasons. Polyclonal preparations are generally easier. These are easier and less expensive. This is the most important point. These are less expensive and used to generate like less expensive than monoclonal antibodies, and they are also able to withstand greater variation in temperature and pH. And in medicine, most of the common use of the polyclonal antibodies confer passive immunity to certain diseases. The only effective treatment for Ebola, for example, is transfusion of serum antibody from a human survivor of the virus. In diseases such as Ebola, this is effective because the virus multiply and act as quickly within the body that immune system do not have time to mount on its own. When somebody becomes infected with Ebola virus, he or she dies long before the immune system can combat the virus. The only effective treatment is therefore the protection conferred by the antiserum. of someone who has encountered the infection previously. Another medical use for the antiserum is antitoxin or antivenin. These preparation contain antibodies specific for venom from poisonous reptiles, arachnids, and insects. They are used to treat people who have been bitten or stung by these animals, again because the venom acts too quickly in the body for the immune system to mount. The idea of the magnetic bullets was first proposed by Paul Ehrlich, who at beginning of the 20th century postulated that if a compound could be made that selectively targets a disease-causing organism, then a toxin for that organism could be delivered along with the agent of selectivity. Now, Nobel Prize in Medicine basically goes to Professor Niels Kejern, Professor George Kohler, and Professor Caesar Milstein for theories concerning the specificity in development and control of the immune system, the discovery of the principle for production of monoclonal antibodies. Now, what are the monoclonal antibodies? Monoclonal antibodies are identical immunoglobulins. So, what are monoclonal antibodies? Monoclonal antibodies are identical immunoglobulins generated from a single B cell clone. These antibodies recognizes unique epitopes or binding sites on a single antigen. Derivation from a single B cell clones and subsequent targeting only of single epitopes. This targets basically single epitope. That's what differentiate monoclonal antibodies from the polyclonal antibodies. Therapeutic monoclonal antibody acts through multiple mechanisms such as blocking of target molecules, functions inducing apoptosis in the cell, which expresses target by modulating signaling pathways. Antibodies can also be used to purify their target compounds for mixture using the methods of immunoprecipitations. Now, how production of monoclonal antibody takes place? We use Hybridoma technology. Basically, these are produced by the Hybridoma technology to produce large amount of monoclonal antibody. Hybridoma technology is a method for producing large number of identical monoclonal antibodies. So, here we can see monoclonal antibodies produced in a plasma cell. So, extracellular antigen comes and B cell is there, and B cell produces plasma cells, and these plasma cells basically produces antibodies. So there is suppose there is an antigen, this antigen and there is a plasma cell inside the mice. So lifespan is short, then artificial culture, and then will produce antibodies. Now, what is hybridoma technology? The plasma cell antibodies combines with the myeloma cell, that is cancerous cell, and antibody plus immortal cells are there, and this immortal cell, plasma cell antibody, myeloma cell combined to form hybridoma cells. Then antigen, then there is a procedure, intravenous injection and spleen removal or isolation of the plasma cell, and this plasma cell inside inside the spleen cell, then infection of B cell, this B cell produces plasma cell, and this produces the antibodies. Now fusion of this plasma and myeloma cell. These plasma cells basically combines with the myeloma cell with the help of polyethylene glycol. This is plasma cell, this is myeloma cell, and this there is fusion, and we'll get some fused myeloma cell, fused plasma cell, unfused plasma cell, and then unfused myeloma cell. But our target should be a hybridoma, combination of both plasma and myeloma cells. Now there is HAT media, that is hypoxanthine aminopterin thymidine media. Then is mother cell, and there is a daughter cell. This is grown on HAT media, and there is this HAT media only follows the salvage path.